Good morning, students. Today we are going to learn about the common histological stains. Common histological stains. First one is hematoxylin. Hematoxylin, from where we are obtaining this hematoxylin? The source is from a tree. Which tree? South American logwood tree. Hematoxylon Leguminosae family. From this tree, we are synthesizing the stain called hematoxylin. Then, what are its properties? It can give blue to black color to chromatin. To what structure? To chromatin. Which color it can impart? Blue to black color. Blue to black color to chromatin. And one more thing here, it cannot directly bind with the tissues. It needs the help of another substance. That's helping substance is called mordant that we already have learned, and it need the help of a mordant to bind with the tissues. Here the mordant used is alum. And next property is pure aqueous hematoxylin cannot stain anything. But actually the staining compound is called hematin. It is the oxidation product of hematoxylin. Okay. Then we have learned the source and its properties. Next slide. Different hematoxylin preparations. Which are the different preparations of hematoxylin? Hayden-Haynes iron hematoxylin one type of hematoxylin preparation. Then Ehrlich acid hematoxylin another preparation. Delafield's hematoxylin another one. Harris alum hematoxylin another one. Then iron hematoxylin. These are the different hematoxylin based preparation used to stain the chromatin. Then about the preparation of iron hematoxylin. Then this iron hematoxylin is usually used to examine the detailed aspects of cells and tissues. For preparing iron hematoxylin, we need iron alum 3% in water. Then hematoxylin 5% in 96% ethanol of its 1 volume. Then distilled water 9 volume. This should be the composition for preparing the iron hematoxylin. Next, another type of stain called eosin. What is the significance of eosin? It is usually used as a counter stain to hematoxylin. What is meant by a counter stain? Counter stain means what? In double staining and in multiple staining, we have already heard that term counter stain. Counter stain means in that staining mixture there will be one principal stain, and this counter stain give a background. The color of that background will give a contrast to that the color imparted by the principal stain. Understand? So that's why it's called a counter stain. So eosin is often used as a counter stain to hematoxylin. Hematoxylin actually give blue to black color to chromatin. So it usually gives pink or red color. So then can clearly visualize the structures that is stained with the blue in a pink or reddish color. So usually it stains cytoplasmic material with a pink or red color, cell membranes, red blood cells. For red blood cells it gives a deep red color. And also it can stain some extracellular structures. Then there are some other stains also there similar to eosin. They are called eosin Y and eosin B. Eosin Y is slightly yellowish and eosin B is slightly bluish tinge. Two dyes are interchangeable because we can use both for we can both these type of stains in similar situations. And there's no matter if we in a way change eosin Y for eosin B or eosin B for eosin Y. No problem for that. So two dyes are interchangeable. Next is carmine. Carmine and is a stain. Its source is from an insect, female coccineal insect, Coccus cacti. It lives on Opuntia coccinellifera. It's a cactus-like plant. Cactus means കള്ളിച്ചെടികൾ. On that plants, these insects are living, and from this insect we are preparing the carmine stain. Major component of this stain is carminic acid. The main properties of the stain: it is a red dye, and also this dye also requires the assistance of a modern, usually aluminium, for binding with the specimen. Example for carmine stain are borax carmine, alum carmine, and acetocarmine. So we are going to see about each type of carmine stain. First is borax carmine. This is a routine stain for hormones. What is hormones? That also we have learned. What is hormone? Hormones means it's such type of mounts in which sometimes we are mounting the entire organism that will be of very micro in size in the organism in one's life. Or otherwise we can mount an organism that is thin enough to transmit light through it. Such type of mounts are called Hall mounts. I'm only remembering you that what you have learned in the last class. And this preparation: caramel 3 gram needed, borax 4 gram, and distilled water 100 ml. And all these need to boil for 30 minutes. And after 30 minutes boiling, to cool it and add equal volume of 70% ethanol. Then filter it whenever we are in need to use it. An important feature of this stain is it is a transparent stain. Transparent means it can permit the passage of light. And next carmine stain is acetocarmine. What is its purpose? It is mainly used to visualize chromosomes in mitotic and meiotic phases. Mitosis and meiosis are two different types of cell divisions. Mitosis is mainly for growth, meiosis for reproduction. Then how this acetocarmine is prepared? That preparation is as carmine powder 1 gram, acetic acid 45 ml, distilled water 55 ml. This all mixed together and heat on water bath just to boil. Next slide. Then Bismarck brown is another carmine stain. Not acetocarmine, carmine stain. Preparation: we need Bismarck brown 2 gram, alcohol 70% alcohol, 100 ml we need. This purpose to stain cellulosic cell wall of the plants, and also mucin that is present in the cell wall, substance secreted by the epithelium. Then next, another type of stain, that is vital stains. Vital means stain that can give color to living cells. Significance also, that's one, to stain living cells. And the purpose to detect cell structure and specific metabolic reactions. Is a property of vital stains. They are lipophilic. Lipophilic means it is able to dissolve in fat soluble. So it can very easily penetrate the cell membrane of living cells. For using it, we need to use a very dilute solution. Concentrated solution may turn toxic to that cell. Examples for vital stain are Janus green bee. It is green in color. Green color in oxidized state, and when it is reduced, it changed to colorless. It is used to study mitochondrial activity during cellular respirations. Other examples are neutral red, methylene blue, trypan blue. Next slide, blood cell staining. Common dyes used for staining blood cells are Wright stain, Leishman stain, Giemsa stain. And Wright stain and Leishman stains are, we are commonly used for our practicals for staining blood cells. That you will do it in your sixth semester, say sixth or fifth semester. I think Wright stain and Leishman stain. These stains are, we are commonly used. And these stains, what is this stain? What is its composition? They are a mixture of basic stain, methylene blue, and an acidic stain, EOC. The mixture of which all stains? A basic stain, methylene blue, and acidic stain, EOC. And they mixed together and dissolved in methanol. And this mixture and based on its different compositional change, preparing this different types of stains, Wright stain, Leishman stains, and Giemsa stain. The nucleus, we know it's acidic because of the presence of nucleic acid, so it has affinity to basic dyes. Cytoplasm is basic, so it has affinity to acidic dyes. So in Wright stain and Leishman stains, this basic stain and acidic stain is present. So it can stain well the blood cells. Then among the blood cells, eosinophils. Eosinophil is a type of WBC. Eosinophil have high affinity to eosin dye due to the abundance of eosinophil granules in the cytoplasm of eosinophil. Eosinophil cytoplasm contains so many granules. That granules are called eosinophil granules. Because of the presence of numerous eosinophil granules in its cytoplasm, the eosinophil WBC have high affinity to eosin. Then next is Wright stain. Wright stain, this one is a common blood cell staining stain, and in which all situations we are using Wright stain to stain peripheral blood smear for making blood smears. Blood smear is actual practical that you will done in your fifth semester. No, sixth semester. And also bone marrow aspirates for examining the bone marrow contents. Then to stain chromosomes to diagnose whether that person have any syndrome or disease. For the differential count of blood cells to understand to count the different types of blood cells. For that also we are using Wright stain. Who developed this Wright stain? James Homer Wright in 1902. How is prepared? Wright stain powder 20 mg, then acetone-free methyl alcohol 1 gram, and dissolve it at room temperature. Next is Leishman stain is developed by William Book Leishman. Ingredients which are methylene blue and eosin. What is its purpose? To stain blood for malaria and other parasites. To check whether that person's blood contains parasites for malaria or any other any other type of parasites which can cause disease, any sort of diseases. For checking that we use Leishman stain. Then Giemsa stain. Giemsa stains preparation. How? Giemsa's powder we need 3.8 gram and methanol 250 ml and dissolve this in water, and to this add 250 ml of glycerol. This is the stock solution. Before use we need to dilute this one. With this we can finish today's section. Thank you.